Thanks, Lynn, and thank you for all the time and effort you spent to get all the awards stated out because it certainly has taken a bit. So I'd like to welcome everybody. Tonight's speaker is David Lewis, MS, is a retired chemist and avid mycologist with a BS and MS from my alma mater, Lamar University in Beaumont, Texas, where his master's thesis was based on the study of East Texas mushrooms. But David is currently a research associate with the Field Museum of Natural History in Chicago, Illinois, where 5,000 collections of his fungi are currently located. He's an honorary staff member associated with the Tracy Herbarium at the Texas A&M University. From 2016 to 2018, he had been the fungal twig, which is the coordinator for the mycologist for the Big Thicket National Preserve all-taxa biodiversity inventory. He's president of the Gulf States Mycological Society since 1998. He's authored many papers related to mycology and discovered several new species of mushrooms. 4 are actually named after him. In 2009, he received the North American Mycological, I have a problem saying that, Association's award for his contributions to the amateur mycology. And in 2010, the R.E. Jackson Conservation Award from the Big Thicket Association. Along with Alan and Arlene Bessette, he's co-authored Mushrooms of the Gulf Coast States, which he's going to talk about tonight. That's a field guide to the mushrooms in Texas, Louisiana, Mississippi, Alabama, and Florida. He and his late wife, Patricia, live on 60 heavily wooded acres up in Newton County, Texas, where they study plants, fungi, and wildlife. So with that introduction, I will turn it over to you, David. Yeah, we see you, and then you want to do the share?
Share, okay. Share a screen. Oh, there we go.
And while he's bringing that up, I want to remind everybody that you can use either the Q&A or the chat to post questions to David, and he will take those at the end of his talk. So.
Okay, how's that? Is that working?
That's good.
Oh, okay. Well, here's the title, Mushrooms of the Gulf Coast States, which I've been working on since the early 70s, and I became interested in mycology when I was a chemist at Lamar and switched over to biology because the taxonomy of Gulf Coast fungi is still pretty well unknown. So it was quite a challenge to start. A little bit more, basically I'm trained as a chemist, but also I have a background in biology also. So it's kind of a sort of a hybrid between the two fields now. One of the first things people always ask me about edibility, and I guess it's human nature to want to know if it's edible or not. Basically, I'm a taxonomist, and edibility is sort of second concern to me, but people always ask me. So we'll talk about some of the morphology, how to tell them apart, and a little bit about edibility. And like I say, certain species can kill you, so you have to be really aware of what you're picking if you decide to eat them. And I would tell people every mushroom is edible once. So that in mind, let's go into a few definitions. First of all, mycology is a branch of science with fungi, the study of fungi. And a mycologist is one having knowledge of fungi. At one time, mycology was a branch of botany, but biologists being biologists, we looked at these things in more detail and decided they have their own kingdom now. So they're in the kingdom of fungi. It's a large kingdom. We have no idea how many species are out there because we keep discovering new ones every year. This illustration shows some of the morphological diversity of the group. And some of the sobering numbers. This is based on a study by a British mycologist, Hawksworth, in '91, where he compared the number of fungi to the number of plants in Britain. He came up with a ratio of 6 to 1. And based on this, he postulated there might be as many as 1.5 million species of fungi out there. Actually, there's some people think it's even more than that. Another thing to look down at viruses. You know, we've been aware of viruses here lately because of the COVID, but we really don't know much about viruses either. So that's something to really reflect upon when you think about nature and the nature of diseases and so on. And so on, we have about 1,400 named species of the Pseudomycetes. That's the more prominent mushrooms that are known from Texas. A little bit about nomenclature. This is sort of it's breaking down how it's broken down. But for all purposes, when we talk about it, just worry about the genus and species. That's sort of your handle on when you're naming species. This is the nomenclature of the common mushroom that you buy in the store. Of course, water mushrooms and fungi. So they have no chlorophyll, no vascular system. They reproduce by spores instead of seeds. Now, spore is a mononucleated structure that's usually microscopic. The so-called vegetative phase is a part that is in the ground. It's like a microscopic thread-like structure called a hyphae. If you see a mass of it, it's called a mycelium. And instead of being composed of cellulose, it's composed of chitin, which is a derivative of glucose. And all the mushroom is the reproductive structure of the fungus organism. They have three functions in nature. Some are saprophytic, live off dead organic matter. Some are parasitic, they attack and kill other organisms. And then they have the most interesting group of the mutualists, where they form symbiotic relationships with other organisms, such as lichens and the mycorrhiza. And a few examples. This is an example of a lichen. The lichen is an algae and fungus combined, and this mushroom just happened to be there. It's not part of the lichen. It just happened to be growing among the lichens. And then we have the mycorrhizal symbiosis. This is generally the fungi connected to higher plants like trees and shrubs and such as this. And this, you can see this mycelium in the soil. It's just very extensive. It essentially extends the root system out into the substrate so they can pull in more nutrients and water and such as that. It actually also interconnects other seedlings. You see the main seedling here is interconnected with these other two seedlings. And this is like a network in the soil that transports nutrients between trees and other organisms. And if you look in the soil, you'll see these sort of swollen structures, the mycorrhiza. This is the actual symbiotic connection between the fungus and the plant. Okay, first I'm going to talk about the Basidiomycota. This is the ones most people are aware of. This is the ones you buy, you buy in the store. You see a puffball, that's a Basidiomycota. Basically, what defines them are they have a structure called basidium, which generally has 4 spores, and these are shot off for the most part. A little bit about the anatomy. This is just sort of a typical structure. You have a pallius or cap. You have a stem or stipe. Some have gills. Some have an annulus or skirt, and others come from this structure in the ground called a vulva. This sort of protects the mushroom as it develops. One of the important things to do when you collect for either the table or for taxonomic research is try to get all the different structures as it ages, because weather and other environmental factors will change the appearance and the color of these things with age. So the more developmental stage you have, the better it is for taxonomic and edibility purposes. One of the first things you want to do is make a spore print. And in the old classification, spore print was really important in telling it how to tell the different families apart and such as that. They have spore prints that range from white to green to black to rusty brown to yellow and so on. And here, usually you just sit with a cap, put it on a piece of paper, wait 24 hours and see what kind of print develops. A little trick I learned is to put it on, let's see, next one. Put it on mylar, pieces of mylar. So you can actually hold the mylar up to either black paper or white paper to discern the color. Here you can see the upper one is sort of a cream color. The one in the middle is white, and the one down on the bottom is sort of a rusty brown. So this, you can see this really helps to get it to the right family or the genus. Now the first group, you want to talk about are the agarics. These have what's called radiating structure, plates on the bottom, the gills. And this is where the spores are produced on the basidia. It's a large group. This is the group I mostly work with. And we will talk about the Amanita family first. This is probably one of the most loved and hated of all genera because they have some of the best edibles and also some of the most toxic mushrooms. And here you can see the typical vulva at the base right here. And here's part of the warts that sometimes remains on the cap. And one of the things I always stress to people, if you work with amanitas or amanitas, you want to make sure you get the vulva out of the soil. Sometimes it's buried several inches in the soil, like this one right here. And without the vulva, it's sometimes hard to get identification if you're doing taxonomic research, or if you're going to eat it, you want to make sure the vulva is not present. These all amanitas do have white spores. So let's remember that. And as I mentioned, some are deadly poisons. This is one of the so-called destroying angels. It's probably one of our more, it's quite common in Texas along the Gulf Coast. It's deadly poisons if you eat too much of it. And we have several of these white amanitas, and they're really hard to tell apart. And here's another group right here. This is probably actually a different species of white amanita, but probably it's also deadly also. Now, here's the thing, being a chemist, I was intrigued with the structure. This is the structure of the toxin. It's actually a protein, a small protein molecule. You think, well, proteins are pretty, you can cook them and they're denatured like an egg. Well, this toxin is quite stable. It's stable to heat. So you can cook the mushroom, eat it, and about 8 hours later, it's destroying your liver because it inhibits what's called RNA synthetase. So it slowly starts destroying your liver over time. So this is, it's quite insidious because you don't know you're being poisoned until 8 hours later. Now, on the other hand, we have one called the American Caesar. This is Amanita jacksonii. We used to call it the European Caesars, but DNA found out that it in North America is different than one in Europe. And interesting enough, the one in Europe is so well known that even the Romans ate it 2,000 years ago. So it's the Americans, the European Caesars is quite well known in Europe. Now the American Caesars is it's a good edible too. You see, it's got the skirt. The bulb's kind of in the ground, but it has over this orange and yellowish cap with the striations. Now, so, well, this is pretty easy to recognize. Well, the thing is, you also have another kind of red cap one called Amanita muscaria, which is a little bit different. You see, it's got more warts on it. The vulva is a little bit different. Sometimes the warts get washed off, so it kind of appears to be the same as the Caesars. This one is toxic and hallucinogenic, so obviously you don't want to eat it. And I always tell people, well, the American Caesars is a spring summer mushroom, whereas the muscaria is a fall winter mushroom. Well, as in science, you can sometimes make mistakes. And I actually found both species fruiting together back in November one year. So you can see the similarities and what you have to be really careful about if you pick these for the table and make sure you're not getting the wrong one. A couple other amanitas. This is one called Amanita rosea pinka. It has these salmon colors right here on the stipe and on the cap. Really beautiful mushroom. It fruits with pine trees in the, in fact, it's fruiting right now with pine trees on my property. Another one, which is a fall amanita, is Polyperamus, a huge thing. The cap is sometimes the size of a dinner plate. It's also found with pine trees, like I say, in the wintertime. And one of our rare, strange ones is one called Amanita westii. If you look closely, it has sort of chocolate colored gills. But the thing is, it has a white spore print. This thing was described from Florida back in the 30s. It was not seen again until 1987 when I collected it over here in Newton County. And since then, we've made several collections, and it's probably one of the rarest mushrooms, although now with more people aware, we're starting to find it in more places. And let's see, we also have another one. This one actually fruits on lawns. Now, most amanitas are mycorrhizal with trees. Well, this is one of the exceptions. It fruits on lawns. It's actually decomposing grass. So it's kind of an odd one because it's a saprophyte, not a symbiont. And this was named for Harry Tears, who discovered it in College Station, Texas back in the 50s. And since then, it's been found in Mexico, and it's slowly moving north. They have found it as far north as Illinois now. So it's with a warmer climate, it's sort of escaping its tropical home and moving north with time. Now, the one that most people see on their lawns, and you probably have seen this too, is the Lepiota, the Chlorophyllum molybdenides, which fruits in these huge fairy rings on lawns. This one has a green spore print, so it's easy to tell. It has a scaly cap, has an annulus, but has no vulva. And you can see you can see the greenish color on the gills. And I get more calls about this, about people eating it, thinking it's something edible, and they get really sick. It has gastrointestinal toxins. So be aware, do not eat this mushroom because it will make you really sick. A couple others in the Lepidoptera family. This is one called Leucocarpinus marmurmii. It's found on mulch, greenhouses, and sometimes in the woods. It has these rather bright yellow colors. Then we have another one, which also has the yellow colors. It's quite fragile, and the name indicates Fragilissimus. So it's usually by noon, the heat has made it fall over and gone away. But it's such an interesting mushroom because it's so small and delicate. Okay, the next family are the Hygropheraceae, sometimes called waxy caps. And these have really bright colors. They have reds, yellows, oranges, greens, and so on. And they're called waxy caps because if you take the gills between your fingers and rub it, it has a waxy consistency to it. And once again, these are also white spored. They have no annulus and no vulva. And wax and also white spored, so it's pretty easy to tell it apart from the other ones. A couple of others we have here, Hygroscope mississippiensis, as the name indicates, is described from Mississippi. We have it in East Texas. And we have Hygrophorus chameleon, which we found in Texas in 2007. And also, it was described from Mississippi. Now, you might wonder why such an interesting name, chameleon. But if you look at the picture on the left, it has this sort of reddish maroon colors. But as it fades out, it turns this kind of dingy looking buff color. What you do, you take the mushroom and you lick it with your tongue, and it reverts back to the original color. So consequently, the name is chameleon. Okay, another one, this is Hygroscope atrosquamosa. It's new to North America and Texas. It was found in the tropics in Trinidad and Puerto Rico. And we found it on lawns in around Houston and Galveston area. And you notice it does turn black with age. Now, next family is Russulaceae, or the genus Russula and Lactarius. These differ because they have they're brittle. All the ones we've talked about before have a rather sort of a rigid structure. They have longitudinal cells, and they're pretty flexible. Well, these are quite brittle. You can throw them against a tree and they sort of shatter. You might ask why. Well, look at the microscopic structure. They have this round cell. It's called spherocyst. So it gives it the stem like a chalky, snaps like chalk. Another feature, the spores are amyloid. They turn blue with iodine. And sometimes the ornamentation, you can see these are sort of spiny. Some are reticulated and so on. So between the round cells and the reticulated spores, you can be sure you have this family. Let's see, we're talking about Lactarius. This is the genus that, as the name indicates, it exudes a latex. You can see the latex right here when you cut it. And we have quite a number down here. This is one called Brolimus. The prettiest one we have, and easiest to identify, one called Lactarius indigo, which we have, we have two forms. We have the summer form, which is more robust. Then we have the winter form, which is more diminutive. So we have these two different varieties that are found in East Texas. Another one we have in the fall is one called Lactarius paradoxus. It has a similar appearance, but the gills are sort of reddish and it bruises this bluish green, and the latex is red. And this one's really nice. It has these beautiful orange gills. This usually fruits around pine trees in the summertime. And as you might figure out, the color of the latex is important. This is one that changes to purple after a while. See, you can see the stains on the purple on the gills. So when you cut it, you almost want to watch the latex, see if it changes color. Some change to yellow, others change to pinkish, others go to purplish. And this is, like I mentioned, Brolimus. So you can see the milk, it just really exudes the milk. has a rather fishy odor, but when you cook it, it's really good. It's probably one of the better ones to eat. Okay, now the sister genus is the genus Russell. Now it does not have the latex, and once again, it has many colorful colors, yellows and greens and so on. Here's one that's sort of a yellowish with this kind of reddish brown cap. This is Russell Metabilis. It has an odor like benzaldehyde or cinnamon. And we have the green cap one, Russell virescens, which the green color, and has sort of like a quilted pattern to it. Then we have one that has this pastel violet color. And as you see, we'll call it Paleocrestosa species nova because it has not been officially named yet. We're still working on this species. And actually, one new to science we found here in East Texas is one called Russell texensis. We found it back in 2009. My colleague from France, who's an expert on this, came over and did some research, and we found this on my property. And he said, well, it's new to science, so we named it for Texas. Another interesting thing about the Russells, they have these red pigments. And I was kind of intrigued by it being a chemist, so I did some extractions and used a black light, and it fluoresces this beautiful indigo blue color. And you can see here on the left, this is without the black light. This is with the black light. So once again, we have quite some interesting chemistry going on here. Okay, the next group are the Tricholomataceae. This is sort of a pigeonhole for everything. It's not the stuff we talked about before, although now it's been broken down in smaller families, but we won't go into that. But just a few examples. This is the oyster mushroom, which is quite a good edible, grows on hardwoods, beech trees, magnolia trees, and oak trees, and so on. And generally, they're more common in the fall. This one is quite remarkable. This is one called Microcybe titans, and this is not a doctored. This is exactly how we found it. This is from Florida, and this thing is huge. It's probably the largest mushroom in North America. And as they say, it's edible. Usually these things are too old to eat. Now, a couple others. This is the honey mushroom, as a name sort of indicates. It has this sort of yellow color. It's a parasitic mushroom. It actually will kill trees. But you can get its revenge. You can get the revenge on it because it's a good edible. It has this yellow color and sort of a ring or annulus on the stipe. Then its sister is one called Armillaria pubescens, which is more brown, and it grows on dead wood. Almost, if you have any dead wood on the ground in the fall, you'll see this mushroom, these huge clusters. You can kind of see the white spore print on the fruiting bodies of the mushrooms underneath the other caps. So you have a ready-made spore print already. Now this is good edible. You have to cook it well because some people have trouble digesting it. Now, just a caveat though, this one grows on wood also, little brown mushroom, but this is in the genus Galerina. And this is one that's deadly, has the aminotoxins like Amanita does. So once again, make sure you do your taxonomy. This one has rusty brown spores. The pubescens has white spores. So word of warning. A couple others. This is some of the decomposed leaves. This is Marasmus Fuvula ferruginous. Has a reddish cap, dark stipe, and you find it on leaves in the summer. In wintertime, we have one called Mycena epipteria. Grows usually on the base of pine trees. If you break it and smell it, it has a odor like watermelon, really a pleasant odor. Then we have a couple others. This was found on one of our projects in the Big Thicket. It was brought in on a mushroom walk in 2006. I sent it on to a specialist, and they did the DNA and compared it to all the mushrooms in the world, and it did not fit the stuff that was from Europe. So we gave it a new name. So this is one called Megacluba texensis. It is found in the Lance Rosier unit over in the Big Thicket. Okay, now we're going to talk about other families that have different colored spore prints. This is the genus Cartinariaceae. The genus Cartinarius is probably the largest genus of mushrooms in the world. they figure maybe 3,000 species in the genus. And it has this rusty brown spore print. You can see the rusty brown spores right here on the stipe. And what this is the veil. This has a veil called a cortina. It's like a spider web. It drops on the stipe like an annulus, and it catches the spores. So right away you have a spore print made for you. So that makes it a little easier to get it to the genus. Let's say it's a large genus. Some are difficult to tell, but this is one we know pretty well. Marylandensis has these really pretty red pigments. There's a whole group of people that love to dye with mushrooms. That's DYE. And they can extract the pigments with different metal salts and get these different colors from the same mushroom. And then, you dye yarn with it. You can make sweaters or socks or whatever. Another interesting one in that family is the so-called corpse finder. This is one in the genus Hebeloma. It actually needs proteins to get the ammonia from to reproduce. And this is it right here. And it's sort of a dull brown spore print. And it's probably quite intriguing because if you have a corpse of decayed meat somewhere, you'll find this mushroom within a year. Another one is the genus Genoplis. This has a rusty brown spore print. Generally grows on wood or decayed pine cones. Then we have the so-called little brown mushrooms. These are small brown mushrooms that generally are hard to tell apart, but this is one we know. It grows, it's the genus Galerina. It grows in sphagnum moss. And then we have the smaller thing, Galerina polyptra, which grows in moss also, and it has a strange distribution. It's been found in England and the Gulf Coast, so it's got a really disjunct population. This one I found across the road on my property in the genus Anosmia. These are called fiber heads because you look at the cap, it has these longitudinal, these radially arranged fibers that radiate from the center. Large genus, they're very important mycorrhizal species. And this one I sent to a specialist, and he says it's probably undescribed. So it's not too uncommon to find undescribed species, even on your own property. Okay, let me take a quick water break. Hold on. Okay. Of course, the genus Agaricus, this is the one you should buy in the store. The genus Agaricus is widely cultivated, mushroom of commerce. It has we can see the annulus right here, no vulva, and it has a chocolate brown spore print. So it's easy to tell it from Amanita. We have a lot of wild ones here. So here's an example. For the most part, if it has a rather pleasant almond-like odor, it doesn't bruise yellow instantly, it's probably edible. And we've tried quite a few and haven't had no problems eating the ones from the lawns. Now, if you're about my age, you remember back in the 60s, there was a lot of interest in hallucinogenic mushrooms. And what brought all this about? Well, there's an article in Life magazine in May 1957. This great adventurers discovered mushrooms that cause strange visions. This was done by banker R. Gardner Watson, who was a banker who was married to a Russian. And of course, Russians love mushrooms, and he found out his wife, would eat wild mushrooms, which horrified him. But he finally, got interested in what's a field called ethnomycology, about the use of mushrooms in cultures. And he became aware of this cult down in Mexico, down in Oaxaca. So he goes down there back in the 50s and does research and finds all these people eating these mushrooms to produce visions. Here we go. Here's some of the illustrations. This is one used by the indigenous folks, and this one also. So there's a whole. This was hidden from the Spaniards for hundreds of years because the Spaniards tried to keep people from doing this. But it's finally people became aware of it again, and it sort of started the the use of hallucinogens for different things. And right now, they're actually finding that the compound is useful for treating depression, alcoholism, and other habits. So if you're under the right circumstances, it can be quite beneficial to as in a medicinal sense. Of course, one of the things you got to be in the right state of mind and have people help you go through these trips. So, I mean, I wouldn't recommend it unless you know what you're doing. This is the one we have quite common on the Gulf Coast. So lots of cubensis grows in cow pastures. It bruises blue. I think you can see some of the blue in right here. But anyway, like I say, I wouldn't recommend it unless you were, were with somebody that knows what they're doing. Well, this is one kind of rare. We found it's known from Alabama, Louisiana, Texas, and Mexico. You can see the blue in color right up here. I've only found this thing twice in 40 years. It's quite rare. The other family are the Antilomas. They have a pink spore print. You can see the spore print right here on the cap of the southern mushroom. And they have the spores under the microscope are angular looking. So they have some really interesting spore morphology you can look at under a microscope. Another one that I just found in Texas a few years ago is this one right here, Antiloma incananum, which grows in around, this was made around Wimberley, Texas a few years ago. It has this really pretty bluish green color, and the odor is like, it says like mouse urine, which is pretty close to being true. Okay, the next family are the boletes. These, now, up to that we've talked about ones that have gills. Now there's others that have tubes. This is the bolete family. You can see these tubes right here. And you can see these tubes right here. It's like a sponge on the bottom of the mushroom. It's just another way to get spores out into the environment. And a lot of these boletes will bruise blue, but it's a different compound than the ones found in psilocybe mushrooms. A lot of these will have these reticulated stems. And this is one of the best we have, Pseudopanophyllus. It's a name indicates it grows on pine trees. One of the best edibles we have along the Gulf Coast. Another one, you have live oaks, which I'm sure you do in Georgetown. This is one that's real common under live oaks along the Gulf Coast called Luridelis. You see the reticulated stipe. It has this brownish cap. Versus red at the stipe. It's probably one of my favorites to eat. Usually you'll find a lot of it when you find them. If you have oak trees, live oaks, you'll probably find this mushroom. It's really quite edible. Okay. Another one we just described about 10 years ago is Boletus rudii, has this red cap, the yellow tubes. We named it for a colleague that discovered it in West Virginia. This thing's known from West Virginia down to Texas and Arkansas. So it's probably common once you start looking for it. Now, there's a sort of a interesting genus called Phyloporus, the gilled bolete. This has the characteristics of boletus, the spores and so on, the DNA. If you look at the structure, they have sort of gill-like structures with this cross-veination. I call this the missing link. Now, on to chanterelles. These are totally different because they have what's called indeterminate growth. They will fruit and keep growing over a period of time. And these are some of the most highly esteemed mushrooms that we have. And the one here, Laniricius, is probably one of the most common mushrooms along the Gulf Coast in the summertime. I mean, literally, you can pick dozens of pounds of it in a short period of time. And here you can see this is some of my property right here. And the young lady is a Swiss mycologist that works with my colleague on chanterelles. They, excuse me, they came here years ago and we described 6 new species over a period of about 10 years. Excuse me just a sec. Okay. One that my colleague described was Cantharellus altipes. You see right here, you see the gill structure, sort of a blunt structure. It's funneliform, no annulus, no vulva, and so on. And once again, all these chanterelles are edible. The one that's really easy is Cinebrinus. It's these red colors. And then we have one that looks a lot like Cinebrinus called Texindus. Morphologically, you can't tell them apart, but you have to measure the spores. The spores are different, the DNA is different. So it just really shows you how much is out there we don't really understand yet. This one we just described about 7, 8 years ago from actually Louisiana, but we have a collection from Jasper County, Gomphus ludovicianus. We had a foray in Louisiana. We found this and we thought we knew what it was. And we just happened to have the world expert on Gomphus with us. He goes back to Tennessee, does the DNA, says, well, fellows, it's a new species, let's name it. So we named it for Louisiana. Ludovicianus means of Louisiana. So far, it's only known from Louisiana and Texas. Okay, I want to talk about a few others, just odd groups. Polypores is a so-called shelf or bracket fungi. They're hard and woody, and they're usually grow on wood. Probably one that everybody knows is the so-called turkey tail, Trimetes versicolor. It has these zonations like here. They have pores on the bottom. You can't really tell because it has a look-alike. This is the false turkey tail, Sterium austria. It has similar banding, but the bottom is really smooth. And these are really important wood decay fungi. Without these wood decay fungi, you'd have wood everywhere. These are the coral fungi, which look like sea corals. And then we have one that looks like a cauliflower, and these things are kind of like strips of bacon. We have the tooth fungi. This is the genus Hericium. You can see they have these long protruding teeth. And this one's a really good edible, and there's not really much you can mistake it for. And then, of course, we have the jelly fungi. These will kind of be gelatinous. They'll dry up, and between rains, they'll dry up and then get rehydrated, so they can keep putting spores out. And we have the puffballs. These are called the gastromycetes because the spores are born internally in a structure that needs a mechanical, like a foot or a raindrop or animal to disperse the spores, whereas the mushrooms do it, by just shooting them off. And this is a large group. We have the genus Calvaceae, which you see in cross section. It's white inside when it's young. Okay to eat when it's like this, but once it starts turning color, it's no good. It goes from white to yellow to a brown to a purple. So then it's not good to eat. But these can be quite common in the summertime. Now, another caveat, cut your puffball in half, make sure it's not the button stage of an amanita mushroom. This is an amanita button right here, which hasn't come out of the egg stage. Another group of the earth balls. These are the genus Scleroderma, which means hard skin. They have purple insides, and these will cause gastrointestinal upsets. And we have another group called the stalk puffballs. They have this gelatinous matrix. They sort of helps the mushroom rise up above the substrate so they can put the spores out. And then we have the earth stars, as the name implies, so they're like a star. And I can imagine if you do a lot of gardening, you'll have these on your mulch. These are the chambered stinkhorn, the genus Clathias. It has usually 3 or 4 of these appendages. And the spores are borne up here in the top, which attracts flies, so the flies can take the spores and put them, carry them to other substrates. And usually you can smell this before you find it. Okay, the last group are the Ascomycota. Up to now we've talked about basidios, which have the basidium and 4 spores. The ascos have this structure called an ascus right here. And generally they have 4 spores that are shot out. So this is how you tell the two apart. Large group, a lot of really interesting morphology. Penicillin is an Ascomycota and so on. A lot of antibiotics come from this group. Of course, yeast are Ascomycetes. This is one of the most important organisms when you think about it. Bread, wine, fermentation processes depend upon yeast. And there's literally hundreds of species of yeast out there. Morels, you should have these around Georgetown because it's, I don't see them in East Texas this far south, but I've seen them around Austin and Wembley and over in around Waco, usually in March. Another interesting thing, this is the genus Hypomyces. It will infect a gilled mushroom, and you can see the slow progression as it infects the mushroom. So it's apparently using the mushroom for its nutrients. It's edible, but you're never quite sure about the substrate. This is an interesting one with Spiralis toxica, new to science just a couple years ago. It's this white fluffy stuff growing on the bottom of this mushroom or this polypore. A colleague of mine works with these things looking for strange chemicals. We found this in the big thicket. He wanted another collection, so I found it on my property, called him up, he came and got it, and we ended up describing it new to science just, like I said, 2 years. The epithet toxica means it's toxic to, he found it's toxic to yeast, to fungi and bacteria. So that shows this many things out there, we don't know what they are. Another one, this is one cordyceps that grows on different types of insects. This is the fruiting structure, and this is the fly. Then we have this one, which grows on truffles. You can see the truffle here, and here's the fruiting body coming off the truffle. And the substrate difference, these two, this is one on magnolia cones, and this is on sweet gum bottles. And this is really neat one. This is Coriaster diaster, Texas devil cigar. This grows on basically along the escarpment from I think around San Antonio to Dallas. It's looks like a cigar. It opens up. And the good news is, guess what? We have a state mushroom. This is a state mushroom. This was just signed as a bill, believe it or not, Monday. So Texas has a state mushroom now. This is it. We do have truffles here. This is Tuber lyonia, which grows with pecan trees. If you've got pecan trees, go out and scratch around the drop line, see if you can find some. And this is an undescribed species from my front yard. And I think that's all. Let's see. A couple of useful field guides. My book, this one just came out with Jay Justice, Amanitas, North America, Mushrooms in the Southeast, and then the Old Texas Mushroom book, which came out in the 90s. And a couple of websites that might be useful. And I think since you have this recorded, you can look this up. And I think that's it.
All right. Well, thanks, David. We had a question there. Somebody was wanting to know which animals, birds, or insects in Texas use fungi as a food source.
Oh, deer, squirrels, turtles. I've seen squirrels eat mushrooms in my front yard before, but deer also will eat mushrooms too. There's a book about mushrooms eaten by deer. Should I just close this out now?
Yeah, you can go ahead and close that if you like, and
Okay.
stop.
There we go. Okay. 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 How's that?
That's good. I am not seeing any more questions right now. So if you have one, you can put it there in the Q&A or the chat. And
someone else knows. It covers all of Texas. Well, it covers mostly eastern Texas, but some of the material you'll find in central Texas also. I mean, obviously, I work mostly in East Texas, so I'm a little, kind of heavily on East Texas material. But you got the Lost Pines over there. You should have it should be good for that. And there's things in the hill country that should be in the book also. Okay, are there mushrooms that are dangerous to touch? Unless you have allergic reactions, I wouldn't worry about it. I've handled the most deadly mushrooms with no ill effects because they won't absorb through your skin. Now, some people could have contact dermatitis. So once again, be aware. Maybe yes, maybe no. Okay, anything else?
Right. What? There's somebody asking, do you use iNaturalist for mushrooms?
Me? No, I don't. No. I'm basically a taxonomist. I make collections. I do the documentation. I take photographs. I do descriptions and dry them out for research. So, I mean, that takes all my time. It's just probably there's thousands of species in Texas. We still don't know what they are.
So if somebody was trying to document them themselves, what would be your suggestion the best way to do that would be?
Well, take good photographs, top and bottom, close up so you can see the structures. If you can, get a food dehydrator, dry them out. If you get serious about it, get a field notebook, take notes, dry them out, and you might find a new species. It could be named for you. You never know. But the thing is, to do the taxonomy, it's quite. You have to really be. It's taken me years to get that point. I mean, you have to really discipline yourself to, sit down. I mean, sometimes I'll spend a whole day writing up collections, because some of these things you have to get really good notes to tell what they are. Because there's a lot of ones that are what's called cryptic species. several species that are sort of hiding under one morphological form, but you start looking at the DNA, the spore morphology, you start seeing these differences. And by the way, I have a collection at A&M now of over 5,000 dried specimens at A&M. So if y'all want to come look at them, you're welcome if I'm in town.
Yeah. So are there any species that require just a DNA to tell them between the two species?
Yeah, unfortunately, there's a few that are so closely related, the morphology. I mean, there's a couple of chanterelles. My colleague from France wrote, they actually, I mean, there's some what's called micromorphology differences, but just looking at them side by side, you cannot tell them apart. But there's differences in the DNA and the spores or whatever. I mean, it's really remarkable how much, and there's also ecological differences we're finding out on some of them.
That's insane. Did you see when somebody asked, they've seen some tiny mushrooms growing out of a wild hog scat. Do you know what mushrooms might be growing out of hog scat?
Oh goodness. Well, it could be a Coprinus, could be Paniolus. These are two genera that are found on scat. Could be a psilocybe too. You know, this sample would be nice. It sounds interesting.
Okay, so several actually grow on scat. That sounds like. Wow.
Oh yeah. Several genera grow on scat, and there's also Ascomycetes that grow on scat too. I mean, it's a whole different. You know, it's another substrate. You know, it's another niche in nature. And once you start looking at these little niches, it's pretty remarkable.
Nature finds a way.
Oh yeah, and of course, you think about it, something's got to decompose something else. So there's stuff that decomposes to decomposers. What are the Crawford dues? Oh goodness, I think 5% maybe. Once again, I'm not a DNA guy, so I can't truthfully answer that.
Are there experts in universities here in Texas that do a lot of that DNA analysis on that, or that mostly done in places across the world?
Let me think. Probably University of Tennessee has a large group. I think Columbia. Let me think. Some in Florida. Berkeley, I think, does some. Yeah, I also up in Washington State. It's not too many people do it. As far as in Texas, not that I know of. I don't think they teach mycology anymore. I took mycology at Lamar and A&M, and they don't teach it anymore. So it's like it's a shame because this is a large group of organisms that we don't have the people to work them up. It's probably 2,000 new species described a year. So you think about that, 2,000 species a year for the last 30 years. That's a lot of things out there we're finding.
All right. Well, I think that may be. Oh no. So yes, there. Oh, invasive. Do we have invasive mushrooms?
Oh yeah, yeah. I mean, yes, sure. Well, I don't know if they're detrimental, but yeah, if you bring trees in from somewhere else, like they brought stuff from Europe to North America, and they had the death cup on the rootlets of the trees they brought to North America. So the Amanita phalloides, the death cap got established on the East Coast. So yeah, sure, there's invasive species.
All right. Well, I don't see more other questions. So thank you again for your presentations. I can't believe that. I didn't know there were quite that many mushrooms out there.
Oh yeah, there's a lot.
All right. Well, thanks everybody for joining us tonight for the presentation. I want to remind you real quickly, we've got our next meeting is July 22nd, and it's going to be Ross Winton, who's an invertebrate biologist from the Texas Parks and Wildlife. And as Sam biologists would say, he's going to be talking about the amazing dung beetle. So thanks everybody for coming, and we'll see you next month. And thanks again, David, for a wonderful presentation.
Thank y'all for inviting me. I enjoyed it.
All right. Good night, everybody.